2020 PRiME Fellows. They will be giving high-level overviews of the collaborative projects that we've decided to fund this year through our PRiME Fellowships. So we will be opening up the polling in Zoom, and we're going to ask you to vote for your favorite presentation. The polls will be open throughout their presentations as well as during the break so that you can enjoy all the presentations before you decide where to cast your vote. And so when we're asking you to view the presentations, again, it's keeping in mind these are 2-minute lightning talks. So we're looking for clear delivery, engaging presentation, and well-organized flow. Okay, and without further ado, I hope everybody enjoys our 2020 PRiME Fellows lightning talks.
Hi, can everyone hear me okay? So I'll start. Hi everyone, I'm Subha, a new PRiME Fellow, and I'm excited to share about my new project on development of broad spectrum antivirals. Next. Every time a new viral infection emerges, we have scrambled for a new treatment. Next. Although the traditional one drug for one bug paradigm has been successful in mitigating viral infections, rapid rate of virus evolution with resistant variants and the increasing diversity of viral pathogens with pandemic potential, including the current COVID-19 pandemic, emphasize the need for broad spectrum antivirals. Can we prepare antivirals to combat the next global crisis? Next. Our project attempts to circumvent these problems by exploiting one drug for multiple bugs paradigm. We aim to target host processes necessary for the virus rather than targeting the virus itself. And since multiple viruses rely on similar host processes, one drug could be effective against many viruses. Next. Towards this goal, we investigated small molecule modulators of host RNA processing and identified 3 distinct compounds with broad spectrum activity against HIV-1, next, adeno, herpes, influenza, and 2 strains of coronaviruses, next. Upon expanding on the observed responses, work at Cochrane Lab has shown that all the 3 compounds modulate host RNA processing and suppress virus replication with minimal impact on host cell viability in vitro. Interestingly, RNA-seq data reveal that Panvir's 1 and 2 R compounds showed limited changes on host RNA expression and alternative splicing events. Next. Now, to position the compounds as lead candidates, I will be merging complementary expertise from Dr. Laposa's team on determining in vivo pharmacokinetics and toxicity of the compounds in mice model. Translating the hits into clinical therapies has potential to impact global unmet medical needs, not only for treating existing infections, but also emerging infections. Thank you.
Hello, everyone. My name is Eric Donders. I'm really excited to share a bit about my project today, which is titled Triggering Release from Colloidal Drug Aggregates. So there isn't any time for questions here, but if you do have one, please reach out to me via the email below. Next. So a major issue in drug development is poor solubility and permeability of potential new drugs. These hydrophobic compounds can sometimes form aggregates that are unable to enter cells, and because of this, they're ineffective. And although this phenomenon has traditionally been seen as a nuisance, we aim to turn these lemons into lemonade and use the drug-rich aggregates to deliver high quantities of drugs into cells. To do so, though, we need a strategy to get the drug to release from the aggregates at the right time. Next. To do this, we've previously used the acidic environment of the endolysosomal pathway as a trigger to get the drug to release from colloidal drug aggregates. We found that drugs that are weakly basic are capable of dissolving in this environment, disrupting the endosomal membrane, and being released into the rest of the cell. However, not all drugs have these characteristics of being responsive to acid. And so for this collaborative project, we will modify a colloid-forming drug with acid-responsive functional groups and examine the effects of these modifications on drug release and endosomal escape. Next. So our first, this diagram summarizes our plan here for this project. In the first step, we start with a drug that aggregates to form colloids and is not responsive to acid. And then through some chemistry, we generate a series of acid-responsive analogs. In the second step, we'll assess these drug variants using a phenotypic assay for endosome disruption. And the immediate outcome of these experiments will be a greater understanding of how drug structure mediates drug delivery through endosome disruption. And ultimately, this approach could allow new drugs to be developed against previously inaccessible targets. Next. So thanks to everyone for listening and to everyone who's helped me with this project.
Hey everyone, thank you for your interest in my project. I'm Brendan, I'm supervised by Gary Bader, and for this project, I'm collaborating with Dr. Alison McGuigan's lab, and we're working on mapping macrophage fibroblast links in hypoxia to try and understand the development of fibrosis. And on to the next slide, please. So the problem with fibrosis is fundamentally it blocks tissue regeneration. This happens when fibroblast activation is dysregulated, which leads to its chronic deposition of extracellular matrix proteins. Inflammation and hypoxia have both been implicated in driving this process, and macrophage-mediated signaling has been suggested as a potential driver. So with that, on to the next slide, we aim to identify druggable targets. What's going on here? We aim to identify druggable targets of fibrosis. And to do that, we're going to use the McGuigan Lab's tracer system to co-culture macrophages and fibroblasts together on this cellulose membrane, which then gets rolled into a 3D structure with a hypoxic gradient. We can then unroll that, collect cells for single cell RNA-seq, and that data gets fed into, if we just push ahead to the next slide, that data gets fed into my cell-cell interaction prediction software, which ranks these interactions on their association with the phenotype, which is in this case hypoxia. On to the next slide. The cool part about this particular collaboration is that because the experimental system is very tractable to manipulation, we can then go back and test our predictions, which sounds like a really obvious thing, but it's quite novel in the context of cell-cell interaction prediction, and look for their association with markers of fibrosis. And that's what I'm the most excited about. So the outcomes are hopefully identifying druggable targets to treat chronic fibrosis, but this is also a wider, more generally applicable pipeline for linking cell-cell interactions with phenotype. And I hope it will also allow me to improve my in silico prediction algorithm so that it can be applied to more difficult experimental systems like patient-derived xenografts where we can't go and test quite as easily. Thank you very much.
Hi, I'm Zhongde Liu. My PRiME project, co-supervised by Dr. Leah Cohen and Dr. Rob Beatty, is focusing on design and synthesis of a fungal selective mitochondria inhibitor as a new weapon to combat systemic fungal infectious diseases. Next. Fungal pathogens, with several major species shown on this slide, are serious threats to human health, causing 1.5 million deaths globally every year. Currently, only 3 classes of antifungals are available for treating systemic infection, and all works through inhibiting fungal cell membrane or cell wall function. Resistance to each has emerged in the clinic. To develop antifungal with novel mode of action is an urgent requirement and the goal of my project. Next. The target we want to inhibit in fungi is a multi-subunit mitochondrial enzyme called BC1 complex. As an essential component of the electron transport chain, BC1 is important for energy production and various processes required for a major pathogen, Candida albicans, to form invasive infection. Next. A promising compound we are working on, which potently inhibits fungal BC1, but not our human enzyme, is an indazole analog, INZ5. In collaboration with the Rubinstein lab at CKIT, we now have determined a hollow structure of alpecin BC1 bound by NZ5. This structure model very well explains many observations from a structure-activity relationship study of the indazole skeleton. In the coming year, with the guidance from this hollow structure, more indazole analogs will be designed, synthesized, and characterized to improve potency, fungal selectivity, and stability. Optimized indazole analog has strong translation potential as a novel antifungal for combination therapies. Thank you for listening.
Good afternoon, everyone. My name is Marius Meidani, and today I will be presenting my work on developing a COVID-19 diagnostic and vaccine platform. Before I get into that, I would like to first acknowledge every principal investigator that already is or will be involved in the project, and they're all shown here. Now let's get into it. Can we get the next slide? As many of us are well aware, SARS-CoV-2 is a coronavirus responsible for the current COVID-19 pandemic. The World Health Organization has shown that more than 35 million people have been infected with this virus thus far, and there are more than 1 million deaths, and currently these numbers are rising. Noting the impact of COVID-19, I wanted to build a COVID-19 vaccine and diagnostic platform. Now, first I'll discuss the vaccine, and then I'll move into the diagnostic afterwards. Next. For the vaccine platform, I'll be doing something very similar to what you may have heard already. I'll be using a vector, which in this case is going to be treated lactobacillus cells to give us these peptidoglycan shells, also known as bacterial-like particles, which are shown on the left of the slide. These particles will then be coated on their surface with a SARS-CoV-2 antigen carrying a peptidoglycan binding domain, and that'll be done in collaboration with Dr. James Rini's lab. We'll then take both of these components and mix them together to get our antigen displaying bacterial-like particles. The goal of this platform is to develop an intranasal spray that will bring these particles to the lung and develop some level of immunity to SARS-CoV-2. In collaboration with Dr. Kozak, we'll be conducting mouse trials on this vaccine platform. Now for the diagnostic, next slide. We are going to be using the same particles and mixing them together with cell-free plasma using a digital microfluidic system, and this will be in collaboration with Dr. Aaron Wheeler. The output for this diagnostic platform is agglutination of the bacterial-like particles via the antibodies in the cell-free plasma. If agglutination occurs, it would suggest that a patient has antibodies that interact with the SARS-CoV-2 antigen on the surface of these bacterial shells. And that is the project. Thank you.
Hello everyone, I'm Jiaxi Peng. My project is analysis of neural stem cell agent by single-cell proteomics. This project is supported by Professor Aaron Wheeler, Cindy Moshead, and Hui Peng. Next. So what population is aging? It causes a series of social health problems, such as neurodegenerative diseases, which need precision medicine to improve the treatment efficiency. For this purpose, it is necessary to study the high-throughput omics and disease biology. This project will try to build a single-cell proteomic platform to investigate the neural stem cell agent precisely. Next. Since the dynamics and heterogeneity of neural stem cells, as well as the low copy number of proteins in a single cell, it is still a big challenge to study the single-cell proteomics. This platform integrates the easy controllable digital microfluidics and highly sensitive mass spectrometry. Also, a series of devices for cell culture, cell lysis, sample preparation, and detection will be designed to reduce the sample loss and improve the detection sensitivity. This is a diverse team. Cindy will give a great support on neuroscience. Aaron and Hui will support expertise in digital microfluidics and mass spectrometry. Next. With the support of this big team, we will try to monitor the single neural stem cell growth, aging, and drug treatment dynamically by our platform. If successful, this project will transform our understanding of neural regeneration at the single cell level. Thank you.
Hi, everyone. My name is Michael, and my project is a collaboration that combines the expertise in nuclear receptor proteomics in the Cummins Lab and the expertise in ligand screening from the Krauss Lab. Next. So this project specifically revolves around nuclear receptor superfamily and its role in precision medicine. And this is a family of receptors that have been proven to be highly druggable targets over the past few decades, quickly becoming the third largest class of drug targets by number of drugs, despite being a small family of only 48 proteins. However, many of these receptors remain orphans with no known endogenous ligands and no therapeutics. So our goal, and the reason that PRiME was interested in this project, is to combine our lab's expertise and come up with a smarter way to look for these nuclear receptor ligands. And considering the success with all the ligands discovered to date, we're hoping that this will similarly lead to the development of new therapeutics. Next. So using the assay that I've developed in the Cummins lab, we can actually quantify these receptors using mass spectrometry to figure out where and when their peak expression is in a group of tissues. And following that with a ligand screening approach that the Krauss lab has developed that we saw from our previous PRiME fellow last year, we can actually look for ligands at these peak times. And we've chosen as our first case to look for ligands in the gut-brain axes. And this is because recent evidence has shown that the gut microbiome is actually capable of producing ligands for these receptors and controlling things like behavior, disease, and metabolism. And due to both the rhythmic nature of the tissues in the gut-brain axes and the emerging interest in this field, we chose to use this as a starting point for our ligand search. Next. So putting this all together, the way that we're going to be doing this is using my assay first to generate a first of its kind circadian protein map of these receptors in the gut-brain tissues, and using that to identify what we're going to call nuclear receptor hotspots. And we're going to screen for ligands extracted from these hotspots. And ultimately, we're looking to then investigate these hits for therapeutic potential, either as new therapeutics to treat diseases associated with these proteins, or investigate disease biology, like under what conditions and why are these ligands being produced. And then lastly, there's also been a lot of success in nuclear receptor therapy by targeting the source of these ligands. And I'm looking forward to sharing these results with you soon. Thanks for listening.
Hi, everyone. I'm Suryashree Subramanya, postdoctoral fellow. I work with Professor Sachdev Sidhu and Stefan Auger on innovative antibody engineering technologies for targeting therapeutically relevant frizzled co-receptors. Next. More specifically, we'll look forward to engineer tissue-specific R-spondin mimics or RSPO mimics as Wnt signaling enhancers. Next. The canonical Wnt beta-catenin pathway starts upon the binding of Wnt to its cognate receptor result along with its co-receptor LRP56, which leads to the stabilization and translocation of beta-catenin, which then binds to TCF, LEF transcription factors to drive Wnt responsive genes. Next. In line with this, our lab had recently engineered FLAGs or frizzled LRP agonist, which can bind to 1, 2, as well as multiple members of frizzled family receptors to stimulate Wnt pathway. Next. In this project, I look forward to design RSPO mimics. R-spondins are molecules that bind to LGR4 as well as ZNRF3, RNF43 receptors. And on doing so, they actually prevent the degradation of frizzled receptors and further stimulating the Wnt pathway. Next. To materialize this project, I will be working with Dr. Sachdev Sidhu and his team, and where I will have access to state-of-the-art protein engineering technologies and advanced antigen display methods like virus-like particles, select-seq-based selections to identify and characterize these CRISPR mimics. Which then, previous slide. So these molecules upon identification would then be evaluated for their effects in intestinal stem cells and organoids from the expertise of Dr. Stefan Auger and his team. Next. So among other potential benefits, CRISPR mimics are going to be a new class of therapeutics for targeted tissue regeneration, including intestinal crypts, bone, as well as liver. Thank you for your time.
Hi, my name is Warren, and today I'll be talking about systematically testing all missense variants for CPOX. Next. Now, next-gen sequencing has become a readily used diagnostic tool, but it's severely limited by the difficulty of distinguishing neutral from functional variants. So many rare, missense variants in disease genes are still interpreted as variants of uncertain significance, or VUS. Now, where there's a faithful assay of gene function available, patients' variants can be tested individually in a reactive manner. Unfortunately, this is both time and resource intensive. Alternatively, scalable functional assays can enable proactive testing of nearly all possible missense variants, even in advance of their first clinical presentation, to yield a comprehensive missense variant effect map, as shown in the bottom right corner. Next. Hereditary coporphyria is conditioned by the deficiency of CPOX in the heme biosynthetic pathway. Because of the associated extreme phenotypic heterogeneity, sequence-based diagnosis is preferred. This creates a need for rapid interpretation of genetic variation in CPOX. Therefore, the goal of my project is to systematically test all missense variation in CPOX. The variant effect map produced could effectively enable more rapid and sensitive genetic diagnosis, increasing the number of patients which treatment can be justified. Next. The framework for variant effect mapping is well established in the Roth lab. It combines codon mutagenesis and large-scale functional assays, multiplexed using next-generation sequencing. Now, preserving genotype-phenotype linkage in a heterogeneous culture is challenged by the diffusion of metabolic intermediates from phenotypically well-type cells to cells deficient for CPOX function. To allow now for multiplex functional analysis of CPOX variants, despite these cell non-autonomous effects, I'll take an interdisciplinary approach, developing microfluidic cell encapsulator in collaboration with Dr. Axel Gunther to isolate individual cells and picolit droplets during growth selection. Next. Now, if the assessment of our model suggests that faithfully predicts damaging variants, the CPOX variant effect map may assist in genetic diagnosis and treatment of hereditary coporphyria. Thank you very much.